Good evening and welcome to the plenary number 3. We have 3 important topics to discuss.
Nous avons 3.
The second about the axis between gut and brain. This is really an important involvement of the research. And the last topic is the problem of the vaccines, not for COVID, but for opioid dependence. In this way, the results for cocaine is not so good. So far, is not so good, but for opioid, is more simple to do. My good friend Amine will present the first speaker.
Thank you, Icro. Merci, Icro. I'm so happy to present my friend and colleague Serge AHMED. Très heureux de présenter Serge AHMED, que vous connaissez évidemment, puisqu'il boxe sur son territoire aujourd'hui, évidemment, il est du Sud-Ouest. Alors Serge AHMED, pour ceux qui ne le connaissent pas, à mon avis, ils sont
Serge AHMED, if you don't know him, he's a researcher at CNRS. He did all his career in addictions. We often ask him to take us about sugar addictions, and it's because of him that the scientific rationale could find its way, because the sugar lobby saying high and loud that there are no sugar addictions. Remember our last exchange, but Serge AHMED is someone who is very well known today. Maybe people won't agree because they're jealous. I think he's one of the experts in the animal models, and we're very happy to hear you in your region. Thank you, Amine, for your introduction. Good afternoon. It's an immense pleasure to be here with you. This is my first Congress in situ. All the presentations I did in the past 2 years were done in front of my screen, my computer screen. So even if there are not many of you this afternoon, it's a great pleasure to have human beings in front of me today. As Amine said, I'm going to talk about cocaine addictions, cocaine dependency, and I will try to talk to you about the research we're conducting now in the Bordeaux laboratory on the loss of control under the influence of cocaine effects, with the idea that even if an individual is in front of several options, when he's under influence, it's like as if his capacity of choice was limited and he behaves as if the only choice was to take drugs. And we will see that at the neuronal level, the mechanisms we are highlighting suggest that the cortical representation of the options is shrunk in favor of the cocaine consumption, but only when you're under influence. Before we get there, we're going to look at something. Someone who's trying to recover, who's trying to get out of the addiction, cocaine addiction, is facing at least 2 issues among many others. The first one is to remain abstinent, to refrain from resuming use. And the second problem is when you stop when you stop, you no longer have that desire, that you resume the use, is to limit consumption when you're under the drug influence. And this is extremely difficult. And today I'm going to talk about this loss of control when you're under influence. And this is like the loss of control concept, which is quite old. It had been defined by Jelinek in the 50s. I remind you, I'm going to speak with my wonderful English accent. In 1950, he said the loss of control is effective after the individual has started drug use. The drug user has lost the ability to control the quantity once he has started, but he still can control whether he will take the drug on any given occasion or not. So in fact, according to that definition, the control capacity is there, but it's lost when you're under influence. And this, there are criteria that we see in the Manual of Statistics of the American US Association of Psychiatrists. When they define addiction, is the criteria that we are not able, once we started consuming drug, you cannot limit the consumption. For instance, I'm going to have just one drink, and finally I drink 2 bottles. I'm going to take 1 small dose of cocaine, and for 2 or 3 days. So this is this loss of control we're talking about, and a study of the different symptoms and the way they interact with each other show that this criterion is at the core, at the center of all the other criteria of addiction. And you find them in many addictions, not only cocaine addiction, but also alcohol, tobacco, cannabis addictions. So the idea is that when you're under influence, something happens in our brain, and we can no longer have a choice. When we have the possibility to choose, but we cannot do it. And this is supported by choice studies that have been carried out in men, in human beings, in the laboratory by several researchers. In these experiments, we have individuals who have the possibility to choose between 1 dose of cocaine or a certain amount of money. And this is a reproducible result. When they have that choice in the lab, the person can go away from the drug and choose the money if the amount is high enough. However, this is when the person is not under influence. If at the beginning of the experiment, the subject is given a dose of cocaine, then the person will choose much more cocaine over the alternative. This is true for a dose of cocaine before the choice study. This was done by Higgins in the 90s. The dose is not a cocaine dose, but a dose of alcohol. And when the person has a choice between money and cocaine, under the influence of alcohol, the person will more often choose cocaine. So you can see that when the brain is under drug influence at the time of the choice goes towards a repetition of the drug intake, as if there is a shrinkage of the alternatives the person has a feeling that there is no other choice. Of course, the question is these neurobiologic mechanisms of the influence, and to find out, we had to use animal models to try to reproduce that situation in animals. What I'm describing here is just it's historical. This started with a choice experiment with the chimps. This experiment was done by Sinistrag at the New Haven lab in 1936. In this experiment, chimps, morphine-dependent chimps, had a choice between morphine or a reward. It was a piece of a fresh fruit, orange, or a piece of banana. And for 80 years, this pioneer experiment was not inspiring, and all experiments on animal models were done in cages. And in the 2000, we were interested again in the choice in animal models, and we were among the first to model this situation. And in this experiment, our animals have a choice, as you can see on this slide, between two levers. If they press on the left-hand lever, they will get a drink, a sweet drink containing saccharide. It will generate a sweet taste that animals like, but it's not nutritional. The animal is not hungry or thirsty. They're just looking for the pleasure of the sweet taste. And the other lever is associated to an intravenous dose of a drug. Here it's cocaine. So if they press on that, they get the IV dose of cocaine. So I show here the protocol. We call that a discrete choice procedure, where every 10 minutes, the animal has a possibility to select between the two levers several times in the session. Why 10 minutes? That's important because 10 minutes is the time necessary for the available dose, for the effects of the dose are dissipated until the next choice, so that the animals are not under the influence of cocaine when they have to make the next choice. It's our control procedure. So the cocaine dose used is very effective. I showed that it has an effect on the measured dopamine. This is the result. When the animals are in that procedure, they can choose between alternative reward that they like and an intravenous dose of cocaine, and they are not under the influence of the drug when they make the choice. You can see that they behave as in the with human beings. The majority prefer the alternative and they don't want the drug. If they are not under the influence, they have the capacity to choose the alternative reward. And this is true regardless of the cocaine consumption history. If they have a few weeks with 2 hours of cocaine, or several weeks with 6 hours of self intake of cocaine. This experiment was conducted. It's another choice procedure, but the Yavincham procedure did in Baltimore. The animals had a choice between methamphetamine in IV or IV heroin, or a brief social interaction with a friend. And every 10 minutes, they had that choice. And even after extended drug exposition, heroin and methamphetamine, the animals, when they had a choice, went away from the drug to have a social interaction with a partner. Whether the partner was someone they knew or a stranger didn't make a difference. This means that the choice is generalized regardless of the option you propose, whether it's a sweet pleasure or the pleasure to interact with a partner. As a rapid summary, we've done many, many experiments with that choice procedure. The results I showed you can be generalized regardless of the drugs proposed, whether it's cocaine, methamphetamine, nicotine, heroin, morphine, or alcohol, synthetic opioids, Oxycodone. It hasn't been done with cannabis yet, but it should be done, regardless of the history of the animals, whether they're naive before the test, whether they have a limited experience of drug consumption or an extended experience developed, if they developed different addiction symptoms. In front of that choice, the animals prefer the alternative reward over the drug. This has been observed regardless of the alternative options, regardless of the sex of the tested individual, and regardless of their age. It was observed among teenagers or adults. At the time of the choice, when the individual, even if they have an experience, several weeks drug consumption experience, when they're not under influence, they choose the alternative reward and they get away from the drug. And that can last for weeks. So this shows that when the individual is not under influence, they have the capacity to get away from the drug to choose another reward. This is when they are not under influence. Now, what happens when the individual is under cocaine influence? To test this assumption, to answer the question, we developed an experience where a few minutes before the animal has a choice, they are administered, unbeknownst to them, an IV cocaine by IV. So it was not signaled. It was not behavior dependent. They were just put under the influence of the drug at the time of the choice. When we did that, we observed that we completely changed the choice of the animals. And when the dose of cocaine increased before the trial, before the choice, the animal changed their preference from the alternative reward to the drug and exclusively a drug choice. What's interesting is the effect of the influence is immediate. There is no learning. As soon as the animal is under influence, there is a change in the preference, and this change lasts as long as you maintain the influence. However, as soon as you stop these injections, passive injections before the choice, the preference automatically goes back to the alternative reward. This shows that the influence of the drug at the time of choice is what determines the choice of the animal in favor of the drug. This is just a variant of the trial to show here the first 8 choices were done when the animal was not under influence, and the next 8 choices after the animals were under influence with a passive IV injection of drug. As you see, the change immediately, so it's very fast. What we try to do is to see at the neuronal level what we can say about the drug influence and that it can be explained neurobiologically. First, this is the work of Dr. KMBM at the lab. She developed, she recorded the electric activity of the orbitofrontal cortex at the time of the choice. This OFC is involved in the decision making and the attribution of values in a choice. So what she did, she recorded the neurons in that area at the time of the choice between an alternative reward and a dose of cocaine. So very quickly, what she highlighted is that the neurons in the OFC are discharged at the time of the decision to go press on the lever to obtain a dose of cocaine or the alternative reward. And she observed 3 categories of neurons. The neurons that become active specifically when the animals choose the drug. Let's call them the cocaine neurons. Others that will become active when they choose the alternative reward. And she also observed neurons which discharge for both options. Let's put them aside for the time being. What's important is that when you look at how these neurons become active at the time of the choice, we see that when the animal is not under influence, it's the neuronal population encode for the alternative reward become active, way beyond the neurons that encode for the cocaine. However, when they're under cocaine influence, we have the reverse pattern. When we look at the activity of these 2 populations just before the choice, there's a suppression of the neuronal activity of the reward neurons, which is more inhibited than the cocaine coding neurons. So what we highlighted also with electrophysiological criteria, we separated the pyramidal neurons from the OFC. Most of the neurons are projection neurons, and inhibitor interneurons. And we highlighted that during the drug influence, when the pyramidal neurons are inhibited and the interneurons are very active, with this idea that interneurons are going to pilot the activity. Okay, 5 minutes of these 2 populations in favor of the cocaine population. We confirmed this result with immunofluorescence experiments. So we marked the interneurons of the orbitofrontal cortex, the GABAergic cells, and we showed that these neurons were activated when the animal is under the influence of cocaine and not the pyramidal neurons. This confirms our electrophysiological analysis, but it's important to confirm that by the immunofluorescence. What we're trying to do now is to take control over the neuronal activity of these interneurons to try. We're developing strategies based on the fact that the pyramidal neurons and the interneurons do not express in the same manner different categories of microRNA. So we're going to use these profiles of microRNA profiles so that the interneurons express ion channels that are light sensitive, so we can either activate them or inhibit them to see if we can simulate the effect of cocaine by directly activating them without cocaine influence, or inhibit them at the time when they are under the influence of cocaine. So that's what we're trying to do, but we're not there yet. But Dr. Guillaume showed that our method works well, so we can express this for light-sensitive ion channels not in the pyramidal neurons. So this is to show you that at the lab we developed these methods of manipulation of the neuronal activity with optogenetic methods. This is an example where we inhibit pyramidal neurons and we see the effect on the choice behavior of the animals. And the first result that it shows that when there's a non-specific inhibition of we favor the choice of towards the drug. To conclude, we finally have been able to highlight that when the individuals are under the drug influence, it interferes with neuronal mechanisms that are important in the choice, with the idea that it shrinks the options available, and the individual has the feeling that the only option is to resume drug consumption. And this, in the long term, for cocaine treatment, I think that what we should target is this influence of cocaine over the choice. That's what I showed. With ideal treatment would be to block the influence of the drug over the choice. And if we're able to do that, the first difficulty, which is to remain abstinent, would not be that important, because even if the individual is no longer resumes drug consumption, if we can control that influence, maybe the individual could limit the consumption and avoid a relapse. That's all. I thank you for your attention.
Is Dr. Comer online?
Yes, I'm here.
Hello. I am happy to introduce to you Dr. Sandra COMER. Dr. Sandra COMER is a professor of neurobiology in the Department of Psychiatry at Columbia University. She received her bachelor's at Vanderbilt University and a PhD at the University of Michigan for her research on the effects of a drug using preclinical models. Dr. Comer is director of the Opioid Laboratory in the Division of Substance Use Disorder, and her current research focuses on the clinical testing of a medication for treating opioid use disorder, methods to maximize the use of naloxone by opioid users, and evaluations of the comparative abuse liability of prescribed pain medication. Dr. Comer served as president of a College on Problems of Drug Dependence, the longest standing scholarly society in the US, devoted to research on substance use disorder, and currently is the public policy officer for CPDD. She joined the expert advisory panel on drug dependence for the WHO and has over 150 publications in substance use disorder. Thanks for accepting to come, and the line is yours. The line is yours, Dr. COMER. You can start your presentation.
Oh, I thought you were going to play the recorded video. Do you want me to do it live?
You can go.
Okay.
Hi, everyone. I'd like to thank the meeting organizers for giving me the opportunity to present our work. I'm sorry I couldn't be there in person, but I hope you enjoy the presentation anyways. So let me share my screen. So I'm going to be talking to you today about some ongoing work that we have on developing vaccines for treating opioid use disorder. This is very much a team effort. These vaccines were developed by Dr. Marco Prevetoni and his colleagues at the University of Minnesota. All of the original research that I'll be presenting today was supported by NIDA, and here are some of my other disclosures. So you may be wondering why we're interested in developing vaccines when we have several other effective medications for treating opioid use disorder and overdose. So I'll go over with you what the clinical advantages are and some of the potential concerns, as well as some of the regulatory hurdles that we've encountered along the way. So the idea is that a series of injections will be given over several months in order to achieve maximal antibody production. So in a person who has been fully vaccinated, if they choose to use oxycodone again, for example, this is the first vaccine that we're developing, is one that's targeting oxycodone, then the antibodies that have been generated will bind the oxycodone and prevent it from getting into the brain. So that's sort of the general idea. We started with 2 vaccines, one that targets oxycodone and its other opioids that are chemically similar, including hydrocodone and oxymorphone. We also are working with this morphine vaccine that targets heroin, 6-acetylmorphine, and morphine. So the idea is that there would be a multivalent vaccine that targets oxycodone, one that targets heroin. We're also developing one that targets fentanyl and its analogs. And then, whatever else comes along could also be added to the list. So just wanted to remind you to get your flu shot. So these are data that were collected in rodents showing how this vaccine works. So along the top panels, I'm showing you the serum oxycodone levels. So serum levels increase and brain levels decrease. This is with a higher and a lower dose of oxycodone. So with both doses, you get these, we get these substantial decreases in brain oxycodone levels. In animals that were vaccinated before training, they never developed oxycodone self-administration compared to the control animals. And in separate studies, we're looking at ongoing oxycodone self-administration. We also see an effect of the vaccine. So in terms of how it fits in the treatment options for opioid use disorder, people often start with recreational use of opioids, often taken orally, that progresses to daily use, and then eventually, may shift to intranasal use, to intravenous use, and ultimately, hopefully the person will be interested in going on to a maintenance medication like buprenorphine, methadone, or naltrexone. And unfortunately, all too often, they will relapse. So a vaccine could be effective in preventing the progression from recreational to daily use of opioids. And it could be targeting, the progression from mild to severe. It could also be used as a treatment medication and as somewhat of a safety net in terms of patients who relapse. And at all of these stages, vaccine could be used for, preventing fatal or non-fatal overdose. So it can prevent the development of opioid use disorder. It can be used as a standalone medication, can be used as adjuncts to other medications. It can prevent relapse if somebody stops taking one of these other medications, and hopefully it can reduce fatal overdoses. So in terms of the pros and cons of the vaccine compared to agonist maintenance or sustained release naltrexone, we know that these two approaches are effective. We haven't yet tested the vaccine here. With regard to transitioning patients onto the treatment medication, it's easy to transition, especially something like methadone from heroin or fentanyl. With sustained release naltrexone, this is one of the biggest clinical hurdles, is that patients have to be detoxed first. With the vaccine, that should be easy because you don't need to detox them. For both agonist and antagonist maintenance, there's a risk of overdose if the patient relapses. Hopefully the vaccine will help mitigate that concern. There's generally good medication compliance with both of the approaches. We're not sure about the vaccine, so that's yet to be seen. In terms of patient convenience, especially with something like methadone, it's difficult because patients have to go to the clinic daily to get their methadone. This may be changing. This is sort of an outcome of the the pandemic, the COVID pandemic. There has been some loosening of restrictions around methadone prescribing. With sustained release naltrexone, patient convenience is good. We're not sure about the vaccine yet. There's one of the downsides of the agonist maintenance is that there's a risk of diversion or misuse. That's obviously not the case with naltrexone, and it would not be the case with the vaccine. Sedation has been, cited as a problem with agonist maintenance, especially methadone, and especially during dose changes. Naltrexone obviously is not sedating, and neither would the vaccine. There's a risk of respiratory depression during dose titration, especially with methadone particularly. There's no depression, no risk of that for naltrexone or the vaccine. There's a child safety risk, and that's not the case with the vaccine or with naltrexone. So one of the concerns that the FDA had was the potential of the vaccine to block endogenous peptides. And so we're measuring that in our study right now. They were worried about the potential of the vaccine to block the rewarding effects of opioids, but not their respiratory depressant effects. We don't think that that's going to be a concern, because if it's just preventing the opioid from getting into the brain, it'll block both effects equally. They were worried about compensatory use of opioids that'll result in increased toxicity. This was also a theoretical concern with naltrexone, but it has not been a real clinical concern here, and we don't think that this will happen with the vaccine either. They're worried about altered opioid pharmacokinetics and pharmacodynamics in vaccinated individuals, so we're measuring that in our study. They also were very emphatic about requiring us to test the vaccine in patients who currently meet criteria for opioid use disorder. So our design is primarily related to safety. So this is a phase 1 study. We are also measuring immune responses as a second aim. And then we're also measuring preliminary efficacy. So we're looking at average peak ratings of drug liking as our primary endpoint for the efficacy. Participants are men and women, 18 to 59 years. They meet criteria for moderate to severe opioid use disorder and are physically dependent on opioids. They're not seeking treatment for their drug use. Their opioid of choice, if it's their opioid of choice is oxycodone or structurally related opioids, we are not accepting them into the study because we don't want to drive them to using something that's more toxic like fentanyl or heroin. if they have any sensitivity, allergy, or contraindications to opioids or any of the components of the vaccine. They can't have acute HIV or active tuberculosis or any other immunocompromising diseases. They can't have previous severe unexpected adverse reactions to a vaccine, including GBS. And they can't be using steroids, antihistamines, or other immunosuppressive agents or other medications within 30 days prior to receiving the vaccine. So this is a mixed within and between subjects design. So each subject will serve as his or her own control. Pre-vaccination and repeatedly post-vaccinations assessments will be made. The between groups variable is the dose of the vaccine. So we'll be testing alum as the control and a low dose and a high dose of the vaccine. We'll randomize them to in a 2-to-1 fashion to either active or control at each of the dose levels. So there will be both inpatient and outpatient periods. And during the inpatient periods, we'll be testing the effects of oxycodone as the target drug. And fentanyl, actually it's heroin, we switched it to as the control. And then lactose powder. So the participants will receive it intranasally. And this study will be conducted clinically at Columbia and also at Clinilabs, which is a contract research organization. So we'll be monitoring both immunological and pharmacokinetic responses. And those data will be collected at the University of Minnesota. So this is what we'll be doing, or this is what we are doing now, actually. So the participant gets a series of vaccinations, every three weeks initially, and then they'll get a booster shot at 18 weeks. We'll have 3 inpatient periods. We'll collect this as a baseline. So this one will be after the 3rd vaccination, when we expect to see good antibody levels. And then again, we'll measure the oxycodone effects at the end. So the inpatient periods involve maintenance on oral morphine, which is not expected to interact with the vaccine response. And as I mentioned, we'll be measuring oxycodone intranasally and heroin as a positive control. So this is what we expect to see. These are data that we collected a long time ago, showing you the visual analog scale ratings of drug liking. All of these medications produce dose-related increases relative to the control. And what we expect to see is that, this is what it will look like before vaccination. And then after vaccination, we'll expect to see a suppression in ratings of drug liking for oxycodone, but no change in ratings of drug liking for heroin. So one of the challenges that we face with this approach is the inter-individual variability in antibody response or the immune response. So there have been a couple of other vaccines that have made it to clinical testing, including nicotine and cocaine vaccines. And for both of these approaches, what they ultimately failed in clinical testing. And we showed that, or the investigators who were examining these approaches showed that in about 30% of the participants who were immunized, they had a good clinical response. So of these 30% that showed a good immune response, we also saw reductions in cigarette smoking and cocaine use. But, but there was just overall, there was too much variability. So some people didn't generate a very good antibody response at all. So one of the things that we're doing in our study is to try to find a biomarker that may allow us to identify participants beforehand who would be a good responder for the vaccine. So, we're concentrating on the B and T cell responses before vaccination, and looking at the effects of the vaccines on these, on these cells, and to see whether or not they're correlated with good antibody response and efficacy in reducing the drug distribution to the brain and also reductions in behavior. So this is kind of what I want to show you some data on that we have now. So these are data that were collected in rodents. So here I'm showing you that the oxy-KLH is efficacious in blocking the ability of oxycodone to get to the brain. So these are brain levels of oxycodone in control animals compared to vaccinated animals. And you can see that there's a fair amount of variability in how well the vaccine seems to be working. So this is, post-vaccination, the antibody titers versus the efficacy. So the ones that have the highest titers are the ones that show the best ability to reduce the oxycodone from getting into the brain. And what Marco showed, and this is really exciting data, is that the animals that showed, oxycodone specific B cell activity that showed the highest levels were the ones that showed the best response in terms of preventing oxycodone from getting into the brain. So, so this was super exciting because maybe we can use this as a measure of likelihood that the patient would respond to the vaccine. So we're collecting blood from opioid naive individuals and those who are diagnosed with opioid use disorders. This is what's being done in Marco's lab and also in a lab in Germany, where I won't pretend that I understand how he does this, but we're measuring B cell frequency and activity here this way. So what we're demonstrating is that, in naive individuals compared to those with opioid use disorders, that the number of oxycodone specific B cells is increased. And this change in activity is correlated with cytokine levels in both opioid naive and individuals and those with opioid use disorders. So cytokine levels are reduced in patients with OUD. And what we have discovered is that the cytokine levels appear to be correlated with heroin use in patients with opioid use disorder. Obviously, is not correlated with opioid naive because these are not opioid users. But for many of these cytokines that we examined, there's a positive correlation. So those with, higher cytokine levels are showing greater uses of heroin. So what we're hoping to see is that, or what we're concluding so far from this part of our research is that there's greater variability in oxycodone specific B cells in current opioid users compared to controls. we've demonstrated that cytokine expression is suppressed in individuals with OUD and that the amount of heroin use is positively correlated with cytokine expression. So we think that B cell activity, cytokine expression, and self-reported opioid use may be useful as biomarkers for predicting vaccine efficacy. And if this ends up holding up, then it could be used as a way of identifying patients before vaccination who may be good responders. So I'd like to thank a lot of people in my lab for helping collect these data. And this is the lab from University of Minnesota, and this is Marco Prevatoni here. So thank you for your attention.
Thank you. Thank you very much, Sandra. Thank you. Thank you very much. Nous avons quelques minutes pour éventuellement poser des questions.
We have a few minutes for a Q&A. I propose that you ask questions to Sandra first because she is quite far away, and then so we don't want to you know, hold a hostage for too long, and then we'll be asking questions to the others. Thank you very much for this very nice presentation. I have two questions for you. You think that there are structural similarities between oxycodone, heroin, and amphetamines, for example. Now, when it comes to molecular recognition, it's not highly probable. So if you want to have a vaccine against oxycodone, you will not recognize heroin, and they will not recognize amphetamines either. So that's my first question. Do you think that there's a structural similarities in between those drugs? Now, what do you think is going to be the duration of action of the antibodies against oxycodone? So in other words, how long will be the immunization period with the vaccine that you aim at developing? Thank you very much in advance for your answers.
I'm so sorry. If you had a question, I would need a translator. I don't have any headphones, unfortunately.
Okay. Just a moment. We are going to translate the question with Rafael.
The first question was that if you suppose that there is taking into account the molecular characteristic of heroin, fentanyl, oxycodone, and enkephalins, and this molecular formula of these molecules is really different, how do you expect that your vaccine will be able to produce antibodies against oxycodone and such a different molecule, such as heroin, enkephalins, or fentanyl? That was the first question. And the second question was, what was the expected duration of the immunization the immunization procedure that you use?
Okay, so thank you. Yes, you're absolutely correct that the vaccine will target a particular chemical structure. So the oxycodone vaccine, based on the animal studies, appears to be effective in blocking the effects of oxycodone and chemically similar opioids like hydrocodone and oxymorphone. And it doesn't produce as good of a response against heroin or fentanyl, for example. So that's why you know, the multivalent vaccine concept is where we're going. We expect to need something like an oxycodone vaccine, a heroin vaccine, a fentanyl vaccine, and you know, now adenitazine is another opioid that has a different chemical structure, and we would eventually have to, you know, make a vaccine targeting that if this approach, you know, if it becomes widely used. So that's what has been the challenge, you know, in terms of vaccine development for opioids, because they all target the mu receptor, but they have different chemical structures. So that's the downside of this approach. The upside is that we can also develop a multivalent vaccine that includes other drugs of abuse. So there's nothing to prevent us from having a vaccine that targets cocaine or methamphetamine and put it all in one multivalent vaccine. So the concept would be similar to a flu vaccine, right? Which targets multiple viruses. And every year we don't know which virus is going to be dominant. And so we kind of make a guess about which components of the flu vaccine will be used that year. So this is kind of similar to that. The second part, what was the second part of the question?
The duration of the immunization.
Yeah. So I assume you're asking how long the antibody response is expected to be. We don't know that yet for sure, but what we're expecting is that it should be effective for several months. At least that's what we're hoping will happen. And we're measuring the time course of the antibody response in our current study for up to, I think, 3 or 4 months after the last vaccine. And so what we're hoping, expecting will be the case is that if, for example, somebody is receiving the vaccinations and they're receiving something like buprenorphine, then we know from the clinical trials that have been done with buprenorphine, methadone, sustained release naltrexone, that the relapse rate tends to be about 50% after 6 months. So if someone had, a vaccine on board on top of that, then theoretically they would still be protected by the vaccine, by the antibodies that are circulating in their system. So we're hoping that it would allow us a window of time to re-engage patients in treatment and potentially serve as a protection against, a fatal or a non-fatal overdose because they have the antibodies circulating. So that's what we're hoping to see. I mean, we'll of course need to do the phase 2 and 3 studies of the vaccine, but that'll be the next step.
Thank you, Sandra. Est-ce que vous avez des questions après
So if you have questions after those 2 excellent questions by Bernard Brock, do you have question for Rafael Maldonado, for example, on the connection between brain and the gut? So you show the transfer of cognitive deficit from man to human, and I thought whether we could translate, more cognitive positive impacts in terms of transfer of cognitive capacity from human to animals and vice versa. Is everything in the microbiota? Not at all. I think it's possible to show a negative impact on memory and on behavior. We start from a basis situation, and it's easy to reduce that initial standpoint. But in order to get a response which improve the result, I'm not saying this is impossible, but it must be more complex, I'm sure. Rafael, I have a question for you. So instead of transferring from the microbiota to a human being or an animal, if we know the process of the different precursors, how they are split, maybe you can have enzymes precursors, because this is an enzyme project. So this would prevent the side effects that you're seeing. Well, the first steps we're doing we identify the microbiota, and then we try to find the different metabolites. And instead of going through the entire process, we can go directly through the metabolites or the enzymes. So I fully concur. Other questions? It's good, Bernard, that you're here. I was very impressed by your concept, the cortical narrowing of the options, of the choice options. In this technique, what is the natural suppression of endorphins when you know that there are reward functions as well? So we tested the influence of opiates when a subject chooses, when they can choose between heroin and a different substance. And you have a different profile. So we tested that with an alternate reward, and there was so when the subject has a choice between a sugared drink or an alternative, we reinforce the alternative, and therefore they do not choose the drug. Now, if we choose very high doses of heroin, doses that we rarely test in animals, but they're very close to the type of doses that a human would use. So when we let the choice to the animal, choosing between a very high dose, and again, they have a continuous access to that sweetened drink, we see a very interesting profile. Animals will take the high dose. For 40 minutes, they're completely stoned. And when they're no longer under the influence, they drink the sweetened drink. They will drink tons of that drink, of that sweetened drink, a lot more than they used to. And once they've drunk, they go back to the high dose and they cycle like that. And at the end, the volume that they drink is 5 times the volume that they would drink in a normal day. And if you test those animals for a long period of time, of course, it has deleterious impact on their body because it's, of course, an excess use of sweet drinks for those rodents. And so when you talk, and that was an observation, I remember I was invited to give a presentation on this experiment with a sweet drink that was in Bordeaux. And I had a number of social workers saying that users under methadone, they would tend to put a lot of sugar in their coffee. So they did see that behavior. So they would not give them too much sugar to those methadone users so that they do not sweeten that drink too much. So it seems that contrarily to stimulants, it seems that opioids tend to reinforce the behavior whereby you choose a substitute. So this is for a sweetened drink. Now, if we think about a choice between a social interaction and a dose of heroin, we know that when users are placed before that choice, some will choose the social interaction, but we do not know whether what they would choose if they would be under the influence when placed before that choice, if I'm being understood. So I think that placed before that choice, they would take a second shot of heroin, but this was never tested. And so, and we haven't checked what's happening at the brain level when they're under the influence of heroin, when they're placed before that choice. Bruno, hello. This is a question for Dr. Ahmed. The question on the decision making. I'm not a learned person in that respect, but you say that the orbitofrontal cortex is involved at different levels. and different cells are activated from what I grasped. And you say that some cells would be more sensitive to cocaine when others would be less sensitive. And the choice of cocaine, if I get that right, will favor the decision-making, one decision-making or the other. So there will be an exaggeration in abuse of substances. So one of my questions in the following: what is the nature of those cells? So pyramidal cells, but what is the nature of those cells? Are we talking about serotoninergic cells, for example? And again, maybe I didn't quite get your explanation. You're talking about interneurons, which are GABAminergic. Okay, so the pyramidal cells use glutamate as a neurotransmitter. Those are projection cells, and we know that in the prefrontal cortex, so in the cells on which we study, they send projection in one of the regions of the striatum, the central ventral striatum. And the function of that part of the striatum is not well known to date, but it is involved in the selection when you have to make a choice, whether you press the left button or the right button. And the decision making, when it's made at the level of the prefrontal cortex, that there are cells that are coding for cocaine, they probably become dominant under the influence of a drug, and they're going to send a stronger signal in the striatum. And this is why it's going to control why you choose to press left and not right. Now, so we know that those two populations are using glutamate as a transmitter, but we have not a finer molecular phenotype for those two populations. So it may very well happen that there are differences at the molecule level between those two populations. And if we were able to identify the molecular signature of the molecules that are coding preferably for a cocaine, maybe we could have targets on which we could selectively act. But to date, we have not reached that level. So we're talking about cellular partnership. So a partnership which would be more or less favorable when cocaine is used versus other partnerships within within the structure of the cortex, right? Yes, this is what we're talking about. And this is why interneurons are important. There are a lot less interneurons than pyramidal cells. However, there are inhibition phenomenon in between both type of cells, which are pretty dynamic. And this explains why a population of cells is going to become dominant compared to the others and makes you choose one action and not the other. And so when you're under the influence of cocaine, and again, this is why we are trying to understand, interneurons would become more prevalent, not prevalent, but would become more important than other coding population. So this is, so far we've understood. Now, how does this really unfold dynamically? We're not so sure. Right, last question, because we'd like to have a break at some point. There's a question for Serge. I was wondering, so we are focusing very much on the impact on the cortex for impulsivity and decision making. And if we inject a dopamine inhibitor, do we have the same sort of effects? So my question is it really the cortical area which has an impact, or other effects of a drug? Yeah, an experience which would be interesting indeed would be to inject cocaine directly in the orbitofrontal cortex to be sure that the influence is really localized there. And indeed, you're right. Whether this is a serotonin mediated action, because we know that this area is quite permeated by serotonin. So we may imagine that if we involve serotonin in the prefrontal cortex, we are preferably going to activate those interneurons I was talking about in my presentation. But then again, we would need also an antagonist, because there are many serotonin inhibitors in that area. And so that would be a whole completely different test and setup altogether. But unfortunately, we have to stop.
Merci beaucoup. Merci Raphaël. Merci
Thank you,
Serge.
Sandra. Thank you, Rafael. Thank you, Serge. Thank you, Icro, for being the co-moderator of that session. I think we're having a break now, and I do invite you as well to take part to the satellite sessions. Thank you very much.